Good. No, well, thanks very much. And I think this is going to be taking a slightly different track, perhaps from some of the previous talks today. But I want to talk about an evolutionary cell biology perspective, because I do think that there's a great deal of value in taking this perspective as we think about the problems that some of the pathogens of humans, particularly the Protistan pathogens, take. And in fact, I want to start just reflecting on my PhD, because it's true. I studied Plasmodium, the malaria parasite, which I'm showing on the right-hand side there. Plasmodium parasite living inside a red blood cell. In this case here, this is actually this little remnant chloroplast that Joel mentioned that I was working on. But in my case, the PhD student sitting next to me was working on plants, and the one behind me was working on algae, and somebody just on the other side was working on amoebae. So my perspective in thinking about these organisms and studying them was thinking about them in this broad context of how do they fit in with the rest of the evolution of life, and in particular eukaryotic life. And indeed, my lab now work on these organisms that I'm showing here. We've got Plasmodium and other apicomplexans. In the middle, we're working on dinoflagellate algae as well. So an algal group, members of the phytoplankton in the oceans and freshwater systems, very important for the health of those aspects of the environment. And we also work occasionally on ciliates as well on the left-hand side there, which again, very important environmental organisms. But you might sort of wonder why are we spreading ourselves across these disparate types of organisms? And the reason is that they're actually all related. In fact, for us, it's a very important reason, because while there are something like 6,000 described apicomplexan parasites, almost all of those are pathogens and parasites of animals, and many of those are very important human pathogens: malaria, toxoplasmosis, cryptosporidiosis, and a lot of important veterinary pathogens. But their closest relatives are the dinoflagellates, this big group of probably 8,000 or so different species. Half of them are photosynthetic, but half of them are not, living in other lifestyles in the oceans and in freshwater systems. So they've got a common ancestor, and of course, this common ancestor is then related to the ciliates as the next most closely related group. And again, a huge group of eukaryotes, very important in the environment, very diverse organisms. And again, I just want to put this in a wider perspective and think about the diversity of eukaryotes in general, because we often do focus on animals and perhaps plants and fungi, but in fact, in this sort of crude or somewhat sort of chaotic tree of life, and it's about as good as we know it at the moment still, but you can see that there's a huge number of groups in this tree of life, and most of those are single-celled eukaryotes. In fact, a lot of these examples, some of the plant pathogens, others are important parts of the environment, microbial communities. So animals are actually a bit of an exception in the global scheme. Again, looking at these organisms that my group work on, the apicomplexan parasites, dinoflagellates, and ciliates, in this wedge over here, even that's just a small fraction of the diversity of eukaryotic life. And if we just look to see where we fit in this scheme here, animals are over here in this somewhat small-looking group, the Bilateria are over here. So we're separated by over 1 billion years of evolution. And this is a really key point, because it means that even though we're eukaryotes, we have a lot of new features, novel features that have developed over that time. Equally, we can look at some other important human pathogens, the kinetoplastids causing leishmaniasis and African sleeping sickness and Chagas disease. They're way over here in this tree of life. Or Giardia, another important gut parasite over here with the diplomonads. Or endomoeba, causing amoebic dysentery way down here, almost the four corners, if you like, of eukaryotic diversity. So we've got a common problem with these types of Protistan pathogens. On the one hand, they're all eukaryotes, so they all share a lot of common biology with us, which means that a lot of antibiotics that are very effective against prokaryotic and bacterial pathogens don't work on these, because we share too much of our biochemistry with them. But on the other hand, we've diverged for a long time, and there's a lot of novel biology that we still don't yet understand in these groups. And this is really the big challenge with studying these organisms as pathogens and trying to think of solutions to them and the diseases they cause. So let's focus on apicomplexans, and that's what my group do a lot of work on. And I'm going to introduce here Toxoplasma gondii, probably the most successful human parasite out there. About a quarter of the global population of people are infected with this parasite at any one time, although thankfully for most of those it doesn't cause disease. But here's an electron micrograph of 2 toxoplasma cells and a schematic on the right-hand side. And you can see it's a regular eukaryote. It's got a nucleus, a Golgi, mitochondria, the sort of things you expect, the sort of things that we also have. I just point out here, there's that unusual relict chloroplast or apicoplast, we call that. This is something that I share with plants. And again, it was a real discovery about 20 or 30 years ago, and very exciting opportunities for drugs for this compartment here. But what I really want to focus on is this novelty. So the apical complex I'm showing here, this is something you don't find in other eukaryotes. You certainly don't find this in animals and plants and other model organisms. And this is key to the infection biology or the invasion biology of these organisms. This is the instrument they use to invade our cells, where they set up residence and parasitize our cells and our tissues. Right at the very apex, there's a beautiful barrel-like structure that's really like a molecular nozzle, in fact. And a lot of these other secretory compartments shown in color here, they secrete through this nozzle. They're delivering invasion factors in a very regulated series of events when these cells infect human cells or other animal cells. So let's watch Toxoplasma do that. And I think it's just an amazing process. This is in real time, a Toxoplasma cell that's sliding in between other animal cells or human cells, and it's just made contact with one, as is shown by the cartoon above. And it's now in the process of actively invading that cell through the plasma membrane. It's making a pore and a constriction as it goes through. And we can't easily see the animal cell that it's going into in this microscopy, but we can see it by the constriction. And in barely 10 seconds, it's been secreting factors into that cell, building an apparatus to invade that cell. And now, in a non-destructive way, it's inside that human cell and ready to start exploiting it, maintaining the cell alive, and it'll be there for about 2 or 3 days. This is about a day later, and that one cell is divided into 8 cells. And this is inside the human cell, human nucleus, just above that there. So tiny cells rapidly consuming that cell, dividing, and ultimately Toxoplasma here will make up to 100 parasites. They'll break out of that cell and hurriedly go out and invade other cells. So an exquisite molecular apparatus and adaptations that allow this process. Exactly the same situation in Plasmodium. So here's Plasmodium inside a red blood cell. In this case here, it's starting to go through division. These cells will break out and infect other red blood cells. And here's another infective form of Plasmodium. This is actually the one that infects the mosquito as the important vector. And again, the apical complex is shown up here. This is that specialized invasion machinery that we find in apicomplexans. So we've got this common feature. It's throughout the apicomplexans. We've got the apical complex, this invasion-related structure. The challenge here is, how do we learn about this? How do we study a structure like this if it doesn't have an equivalent or a homologous structure in other eukaryotes? This is the challenge that we're faced when we try to study these diverse pathogens. So can we turn to genomics for the solutions? And of course, genomics have been a wonderful resource in the last 20 or 30 years for these organisms. Well, we can and we can't. This is, I think, where we see a very interesting picture here, because here we're looking at the genes in common between Toxoplasma, Plasmodium, and in this case, us, the human host for these pathogens. And of these large number of genes that each of these organisms have, there's actually only a small minority that are in common, about 1,300 proteins that are in common between these 3 organisms. These are the proteins that drive these common features, the nucleus, the Golgi, the ER, and things like that. But you can see, in fact, for Toxoplasma and Plasmodium, the majority of their proteins are not shared with humans. And it's almost worse still. We've got about 1,500 or so that are shared with Toxoplasma and Plasmodium, but even these 2 parasites have got about half of their total proteins are novel and not even shared between these 2 organisms. So this is the problem with these divergent organisms. They've spent so long evolving and adapting in a new niche that they've got a lot of novelty that's very hard to study. And if we look at Toxoplasma and Plasmodium and just look at the genes and the proteins that we've got any clue about what they do, about half of them, a third to a half of them in both of these organisms are completely unknown. We haven't got a clue what they do. They don't share any similarity to anything that we see elsewhere. So this is the challenge. Now, I always tell my students to make sure you read the old literature. They don't usually take it quite this literally, but if we go back 2,500 years ago to Sun Tzu's famous The Art of War, one of the points that he made, that if you're going to combat your enemy effectively, and I think we could argue that as parasites and pathogens, these are our enemies, you've got to know the enemy. You've got to know their strategies, you've got to know their arsenal. And I think what this picture shows here, this Venn diagram, is that it's just how little we do know these organisms because of their evolutionary divergence. And we've got to use tools that are available to try to tackle this major problem. So these are the broad questions that I want to sort of pose now, and these are the sort of things that we work on in the group. How do we study a cell that looks like a regular eukaryote, but has so many different genes because of the long evolutionary separation from other organisms like us? Second question is, how do we study and interpret novel structures like the apical complex that are necessary for invasion if they are novel to these groups? And what are the mechanisms by which drug resistance develops? Again, a very important question we've got to ask often with parasites. And malaria is notorious for developing resistance to the drugs that we use to protect ourselves from it and treat these diseases. This is my view of a solution, and I guess I've got a bit of an engineering background, and this is how I like to think about problems, complex problems. A diagram like this for a transmission system for a car, an exploded diagram, it shows all the complexity of this system. It shows you how everything relates to each other. And even though it doesn't tell you exactly how this system works, it's a great place to start to make hypotheses about how different components contribute to the overall function of a complex system. So this is what I think we need for these organisms, because the genome is really just a parts list. And, you know, a pile of parts, where it's pretty hard to interpret that if we don't know what we're looking at, particularly if they're unknown parts. But if we've got some sort of reference of how they all fit together, this, I think, is going to be a great way forward. Now, I'm very, very fortunate to work in the department with a wonderful and brilliant colleague, Catherine Lilly here. She developed a system that I'm going to talk about today, now, and that we've been implementing in our lab, and Constantine has been the chief instigator of this on the pathogens. We're asking this question: what's the cellular context of the proteomic complexity of a cell? You know, I want that exploded diagram, and Catherine has provided the tools to do that. We call it LOPIT. It stands for localization of organelle proteins by isotope tagging. That's not really important. It's just a nice name to use. But this method simultaneously captures the steady state location of thousands of proteins in a cell where they are, whether the nucleus, the Golgi, the invasion related structures for these cells. So I want to quickly talk you through how this works, because this is something which I think we're now applying to lots of different parasites and lots of other organisms across that eukaryotic tree of life. So what Constantine did is he took a collection of these cells, or a population of cells. These are our toxoplasma cells that he did this with. He first of all broke them open under pressure in this case, but he wanted to dismantle or pull apart this cell, but hopefully keep each of those structures all intact. He then dispersed those across a tube with a density gradient. And really what we're trying to do is not purify anything, but allow everything to reach its buoyant density, and different parts of the cell will be denser than others. A good way of distributing different parts of the cell across this gradient. So the next key is that you take fractions across this gradient, and you measure and you quantify the abundance of all proteins in each of these fractions. So we've got a graph of that now, and I'm showing just a single example in black there. This is how abundant that protein is across this gradient. You can see there wasn't much at the top of the gradient, and then there's a long, broad peak of it in the middle of the gradient. So we can use mass spectrometry to make these measurements for many thousands of proteins simultaneously. And in fact, I'm showing those all. They're pretty hard to see, almost 4,000 proteins all in grey in the background there. But we can then say, well, what are the proteins we know something about already? And in this case, I'm taking a collection of proteins that we already knew would be in the mitochondria, and we already knew some would be in the Golgi apparatus, two different organelles of these toxoplasma cells. And I've mapped in brown and green the distribution of those on this gradient. And I hope you'll appreciate that our unknown protein in black shares a much more similar profile to the Golgi proteins in brown than it does to the mitochondrial proteins. And if our Golgi is staying intact across this gradient, wherever the Golgi is, no matter what its concentration, all of the proteins in the Golgi will share the same concentration or the same abundance there. So this is already giving us a good clue that our black protein is in that Golgi compartment as it disperses across this gradient. Now you can do this experiment multiple times, and this effectively gives you more power to your analysis. they're not actually replicates here, they're just different experiments. And you can see in each case, our black line faithfully follows the same pattern as the Golgi rather than that of the mitochondrion. So in this case here, on an individual case basis, we would suggest that this unknown protein most likely is a new Golgi protein that we didn't know about before. But the power of this analysis is that we can ask the same question for the whole cell, for all of these proteins at once. Now, this is now multi-dimensional data, and that's difficult to interpret easily on its own. We can use methods to reduce this to 2 dimensions. So I'm showing a plot here where every dot is one of these toxoplasma proteins, almost 4,000 proteins, and they're aggregated together in clusters according to the similarity of their profile across the gradient. So all of the brown Golgi proteins and this black protein will all come together in a single cluster here in this representation. So you can see there's lots of clusters, and this is indicating that we've got lots of cell compartments with their proteomes all held together there. And again, we can test this. We can say, well, what are the other proteins we already knew about that were in certain compartments, and do they all cluster together according to their compartment? And in color here are those proteins that we already had some idea of which compartment they'd go into. And you can see we are getting clustering together, again, reflecting the same case as the Golgi here. But then we've got all these unknown proteins that I'm showing in gray that are in these same clusters. And then this is exciting because this is the proteins that we had no idea where they would go. And now we're getting strong evidence of them going in these particular compartments. And we can use sort of more mathematical tools to just probabilistically allocate those unknown proteins to these new clusters. And then ultimately, we've done a lot of microscopy where we've tagged some of these new proteins with fluorescent markers. Here are 4 toxoplasma proteins here, and these are 2 new proteins in green and red that are in the apical complex, part of that of the conoid part of the apical complex. So this has really been a wonderful game changer for us with toxoplasma. We were able to, in one experiment like this, over a couple of years, it did take a while to do, but we could allocate 2,500 proteins to one of 26 organelles or compartments within toxoplasma. And we validated a lot of those by doing this microscopy just to make sure that the data really is robust. But as I say, this is now a wonderful tool to start asking questions where otherwise we only had hypothetical unknown proteins. And I want to draw attention now to some apical complex proteins, because this cluster down the bottom here, shown in black and yellow, had 63 proteins that were all associated with the apical complex. And again, these are some of the ones I'm showing up in the top there. And I think this is a really nice aspect of this data, is even without knowing what the function of these proteins is, now that we know that they're there, we can start asking questions about this structure and start using evolutionary inference to learn more about it. And in particular, here I just want to ask this question, well, where did the apical complex come from? Because it's always important to know what the origins of a structure is, to really understand it and understand how it's been deployed in a new function in a cell. So we want to ask this question, where did the apical complex come from? And will this group of 63 proteins help to give us some clues to that? So another scientist in my lab, Ludek Karini, he took on this challenge. He took those 63 proteins. Now there's a cartoon of the apical end of toxoplasma. In particular, he was focusing on this barrel-like structure, the conoid. And this is quite an active mechanical part of this cell. It moves in and out. And in fact, it moves out when it's ready to invade. So it's sort of locked and loaded here. And you've got all of these organelles about to secrete their contents through the conoid here into the human cell as it invades it. And here are 8 parasites here with their apical complex and one of these proteins that Ludek's looked at, forming a ring at the apex there. But he very carefully used high-resolution microscopy to allocate where exactly all of these proteins go. And here are 4 proteins or 5 toxoplasma cells here. Just zooming in at their apical end, we can start to place these proteins relative to each other and relative to these known structures. And he could develop a very detailed model of where all these proteins go in the apical complex, particularly associated with this invasion-related conoid. So then the next question, and Elko Troma got involved in this, is to say, well, are these proteins in other apicomplexans? And what I'm showing on the left-hand side of this rather complicated plot here, but on the left-hand side are all of these new apical complex proteins that Ludek had studied. And then across to the x-axis here, we're looking at all of the other related apicomplexans that we've got genomic data for. So Toxoplasma is on the very left-hand side as part of the coccidia. Plasmodium, all of the Plasmodium species that cause malaria are here, and a number of other apicomplexans through here. And what we can see is that certainly some of these apical complex proteins are just unique to Toxoplasma and its closer relatives. But if I just sort of zoom out a little bit now, you can see there are great big blocks of apical complex proteins that are conserved throughout Plasmodium and other apicomplexans. Now, we wanted to make sure that just because they're conserved, does that really mean that they're also at the same structure? And we teamed up with another colleague, Rita Tawari, and we looked in Plasmodium, and all of these images are Plasmodium cells, and in green are the proteins we've then looked at and verified that they're actually in very similar structures to what we see in Toxoplasma. So it's not just that they're conserved, but they're also doing similar things at the apical invasion-related end. So we do have good conservation of the apical complex right throughout apicomplexa, which we might have expected because they're all parasites. But I think a much bigger surprise was to see that this conservation extends well into the dinoflagellates here, this large block of conserved proteins. So that tells us something which we didn't know, that the apical complex is not just in parasites, it's also in the related dinoflagellate algae. And perhaps that shouldn't be so surprising, and it's certainly very interesting, because here's one of those more ancestral members of this group. This might look like a parasite about to invade this cell, but actually this is a free-living predator in the environment that's eating this cell. These two Colpodalis cells swim around in the oceans. They found an algal cell here. They don't invade it, but they use the same apical complex type structure to latch onto it and to suck the contents of this out into their food vacuoles to feed. So really it tells us that the apical complex didn't evolve for parasitism. It's got an older history, and it's got an earlier use in this feeding type of interaction. And again, this has been a revelation for us in the way we think about the invasion structures. We've now got to think about them not necessarily in the context of evolving for parasitism per se, but evolving for perhaps a similar type of function to make contact with another cell and start to interfere with that cell and originally for feeding. But I think this really is important because it shapes the way we go forward, trying to understand how the invasion machinery works. And equally, we're now working on other members of the dinoflagellates, trying to see, well, what are these structures doing? What are their behaviors and their activities of their proteins? So again, a good evolutionary context to try to pursue these questions. Just finally, I want to touch on another aspect of the work that we're doing and using this more evolutionary approach. In this case, we're addressing the question of what are the mechanisms which parasites use to become resistant to drugs. And again, this is a major problem in combating disease, and artemisinin, which has been our lead anti-malarial compound for about 20 or so years, we are starting to see that resistance is occurring. It's starting to fail in some parts of the malarious parts of the world. What we know actually is we see accumulation of mutations or occurrence of mutations that correlate with tolerance to artemisinin of Plasmodium malaria parasites. And it's interesting it's being rather regional in that in Asia, mutations correlate with a gene called K13, which we otherwise haven't known what K13 does. It's a novel protein that's not in other eukaryotes. And again, we can predict some conserved regions of this protein, but we really don't know what the function is, or we haven't known. However, in Africa, mutations occur in a different gene, in a gene called the AP2mu gene. And actually, this is a gene that my good colleague Joel Dax in Alberta has got a lot of expertise in. So we've teamed up with him for this next part of the story. So we have these two seemingly unrelated, or we couldn't relate these two sets of mutations that we were seeing in Plasmodium. But we again had a look across apicomplexans and saw these proteins are both in, or they're in all apicomplexans. So we came back to our model of Toxoplasma to see if we could use that to leverage understanding of why these mutations that are occurring, whether there's any relationship between these mutations in these two parts of the world and a common mechanism for drug resistance. So Ludic and Brandon in the lab, they first of all wanted to look to see where are these proteins in Toxoplasma. And first of all, they looked to see where K13, this novel and unknown protein is. And it occurs in 2 or 3 spots in each of these cells, rather lovely small rings that actually occur right at the edge of the cell. You can see one at the edge of the cell here. These are 3-dimensional images of these cells. So these are the rings here actually on the surface of the cell. So that's where K13 is. What about the AP2mu protein? So in this case here, we're looking at the AP2 proteins. And again, we're seeing a couple of dots in the cells. Initially, we did these separately, but I think you can already recognize that when we look at them together, we see that these two proteins go to exactly the same structure or the same compartments of these cells. And we had no idea what these were in toxoplasma because we'd never seen these structures before in any other protein locations. So again, using some of those broader tools where we look to see what other proteins are co-locating in the same part of the cell, Brandon and Ludek were able to compile a large list of proteins that are all locating at this peripheral structure in the cell. And Brandon went on using electron microscopy to show that it occurs at these small pits in the plasma membrane of the cell that had been observed for about 60 years by electron microscopy, and they've been called the micropore, but we didn't know what these structures did. And in this case here, we've shown that all of these proteins are located here. And importantly, and as Joel was able to help us decipher, a lot of these proteins are specific to endocytosis. And indeed, this we have now verified that this is an endocytic structure. It's where protein material from outside of the cell is taken into the cell, either feeding or recycling molecules from the outside. So this helped us to illuminate that artemisinin resistance is occurring by parasites reducing the endocytosis or the rate of endocytosis. We now know that this is really a good way for malaria parasites to avoid or to grow tolerant to artemisinin. But it's useful to know this because in actual fact, it just slows the parasite's growth down, and artemisinin only stays active in the body for about 2 hours or so. So if the parasite can slow down and almost hold its breath to get through that drug dose stage, it can go on and survive. But it leads us to recognize that if we change the regime of artemisinin use in these cells, then if we increase the frequency of dosage, that would be one way to combat the resistance that we're seeing. And just finally, I want to just step back again and recognize the fact that a lot of these proteins are common for endocytosis, but some of them are unique to these organisms. Not only apicomplexans, but K13 is also in the dinoflagellates. And actually, dinoflagellates, like apicomplexans, have got a rather armored appearance where much of their plasma membrane is precluded by these plates or these structures. And it looks like the micropore is a little window between those plates to allow endocytic function. And this is something, again, that is common to dinoflagellates as well as apicomplexans. So again, we've got this old history, and we've got to think about these functions in the context of dinoflagellates as well. So, that's just a quick tour of how we're leveraging diversity of cells and recognizing that they've all come from a common ancestor, and they're deploying some of the same machineries for new roles in some cases, but they've got an ancestry that might have been involved in a different activity. And I think I'm going to liken this, just my last image here, to the Rosetta Stone here, because the Rosetta Stone and the ancient Egyptian languages were only able to be deciphered because of the comparison, in this case, to ancient Greek at the bottom. And cells and organisms are just like our languages. They're puzzles, they're codes, they have their own language. And there's tremendous advantage in comparing between them, learning what one will tell us about the other. And often we only really make the key discoveries by having a broader perspective on what these cells are doing and what they're telling us. So thank you very much for listening. I just want to make sure I acknowledge all of the great lab members and collaborators that have worked on these projects, including, of course, our colleagues here at the University of Alberta, and of course funders for providing support for these projects. Thanks.
Great. Thank you so much, Rob. Are there any questions?
I can only just hear you, Joel.
That's because we told them like that. Is that better?
Ah, yes, good.
Great. Okay. Are there any questions? I know there are people in the audience who work on Plasmodium, so I'm going to stare at them until they come up with something clever to say. Ross, maybe I'll start with a question then. You talked about some of the other organisms that you're trying to do LOPET on. So what, I know you've got the Protinsis, but then what else, what are some of the other ones that you're working on?
So we've got some really great data with Plasmodium. So that was the next obvious thing that we wanted to try. We've been looking at Plasmodium, looking at different stages of the red blood cell development of Plasmodium. So we've got some great data there. We've got some also some very good data for Cryptosporidium, and that's a work that hopefully will be released and available through publication very soon now. It's going through the review process well. We've also got some trypanosome data. And again, the trypanosomes have been really fantastic, and we've got really well-resolved, even though they're not so closely related to these organisms, but again, important human pathogens. So the method is working extremely well, and it's been able to, as I said, work equally well in these different organisms that we're working on. We do have dinoflagellates as well, a couple of dinoflagellates, and as you say, Perkinsus as another related parasite to the apicomplexan parasites. So it is a great tool, and I think, in my mind, I think this is sort of the next phase. We need to take a lot of the genomics work in, particularly for these organisms that are so distantly related to the models like yeast and mammalian cells that are otherwise better studied.
Fantastic, great.